Welcome students. Today we will talk about nodal anatomy. The vegetative plant body of the sporophyte of a vascular plant consists of three main organs, that's stem, root and leaf. The demarcation between the stem and leaves is particularly uncertain, and therefore the shoot is a collective term for the stem and its appendages. The stem is divided into nodes and internodes, and one or more leaves may be present at each node. Besides leaves, buds also occur on the shoots. Depending on the level of development of internodes, the shoot assumes varying heights. Thus, a common feature of the stem in the primary state of development is its division into nodes and internodes, which results from the manner of origin of leaves at the shoot apex and the subsequent growth of the axis bearing these leaves. These features of the shoot have a bearing upon the primary vascular system and its development. The primary vascular system may have a shape of hollow cylinder, or it may be divided into discrete organized strands called the vascular bundles. The type, size, number and arrangement of these strands vary amongst the seed ferns and vascular plants. These variations in the structure of primary vascular cylinder are obvious while we study the anatomy of nodes, where the central cylinder is broken by leaf gaps and branch gaps. Nodal anatomy is considered as one of the most significant aspects of angiosperm systematics and of the comparative morphology of stem, leaf and flower. The detailed histology of the vascular bundles may be different at a node, a feature particularly determined by the lack of elongation. The cortical and the pith cells may be shorter. There may be less sclerenchyma and more collenchyma than in the internodes. The degree of differentiation between the nodes and the internodes is influenced by the relative development of leaves attached to the nodes. If the leaves are rudimentary, as in underground stems, nodes and internodes differ little from each other. Thus, the development of vascular tissue is intimately associated with leaf formation. Nodal anatomy accordingly describes the pattern of vascular connection between the leaf and the stem that is maintained by vascular bundles called leaf traces, which are associated with parenchymatous interruptions in the stem vascular cylinder called leaf gaps. Now, leaf traces. Prolongation of the stellar vascular tissues at nodes that supply the leaves constitute the leaf traces. The trace that supply one leaf constitute the leaf supply. The term trace is applied to these bundles from the point where they first become evident as foliar supply bundles to the base of the leaf. If the vascular bundles diverging into a leaf are traced backward into the stem, they may be found to be discrete for variable distances within the stem, and then ultimately to merge with other parts of the vascular system of the stem. A trace may be pictured as extending between the base of a leaf and the point where it is completely merged with other parts of the vascular system in the axis. A number of leaf traces in various plant groups. The number of traces supplying a leaf varies from one to many, and this number is usually constant for a given species and often for a family and even a larger group. This number is the number of bundles that leave the stele. These in course through the cortex may fuse or branch so that the number entering the leaf is different. In the pteridophytes, there is usually one leaf trace, though sometimes there are two or many. In gymnosperms, there are one or two leaf traces, while in angiosperms, the number of leaf traces may be 1, 3, 5 or many. Of these traces in angiosperms, 3 is perhaps the primitive number. Where there is only one, this is either the result of fusion of the original 3 in evolutionary development or of reduction of the 3 to 1 trace by the loss of the lateral bundles. 3 and 1 is the most common leaf trace number in the flowering plants, 3 being characteristics of nearly all the Amentiferae and of such families as the Rosaceae, Aceraceae and Compositae. 1 leaf trace is characteristic of the Lauraceae, Ericaceae and Labiatae. The number of leaf traces is independent of the size, type and duration of the leaf and of the nature of its attachment. Many plants with large leaves such as Fraxinus have 1 trace. Others such as Juglans have 3 traces. Still others such as Aralia have many traces. Likewise, plants with small leaves have only 1, few or many traces. Now, arrangement of leaf traces. The arrangement of leaf traces and their complexes varies in different groups of plants and is related to the phyllotaxis. The phenomenon is well understood when we study the vascular bundles in a stem. Stem vascular bundles are commonly classified into first, axial bundles, second, leaf trace bundles, third, branch trace bundles and fourth, sympodia. Now, an axial bundle is also called colline bundle or common bundle. They form the major vascular bundles of the stem and are not restricted to any one leaf or branch. By anastomosing, they may form leaf and branch trace. A leaf trace is a bundle that diverges from an axial bundle or another leaf trace and extends into a leaf. Branch trace diverges from the stem into a lateral shoot. A sympodium consists of an axial bundle and its associated leaf and branch traces. In some plants, the leaf trace forms sympodia that are independent of one another. In others, the different trace complexes are interconnected. In monocotyledons, the common plant is the so-called palm type, where the leaf traces of a single leaf can be roughly divided into small and large. Now, departure of leaf traces from vascular slender. The trace of leaf normally depart individually, often at considerable distance from one another, laterally about the stem as well as vertically on the axis. Frequently, the trace departs side by side. Traces most frequently depart from that segment of the stele which lies directly beneath the attachment of the leaf. Traces may depart even from the side of the stele opposite to that on which the leaf stands. Such traces enter the base of the petiole directly if the latter clasps the stem extensively or may girdle the twig in the cortex, swinging around the stem as they pass upward through the cortex to the petiole base. Such traces that pass around the stem for some distance in their course from vascular slender to petiole are called girdling traces, for example in cycads. The angle at which the trace leaves the central slender varies greatly, being usually very small. The strand departing gradually from the pith and passing obliquely, sometimes almost vertically through the cortex. Less commonly, the trace passes outward at nearly right angles to the stele, entering the leaf base after a very short course through the cortex. The number of bundles at a given level of the stem is determined by the phyllotaxy, the number of axial bundles, the number of leaf and branch traces, and the number of internodes the traces traverse. There may exist certain changes in the number of bundles that occur along the stem of one plant. For example, in the cross section of the stem that develops first, that lowermost portion, the number of bundles is less than that seen in the cross sections of higher portions. A further decrease in the number is also seen in the portion prior to the development of floral primordia. Now, leaf gaps. In the majority of vascular plants, the outward passage of a leaf trace is associated with the differentiation of parenchyma in the vascular region of stem. instead of vascular tissue around and above the point of departure of the trace. This opening or break through which the cortex and pith becomes continuous is known as leaf gap or lacuna. Leaf gaps are constant in appearance in the great group of vascular plants known as Chiropsida. Leaf gaps are absent in Lycopsida, a group which includes club mosses and a few other similar plants. In plants with vascular system composed of anastomosing strands like certain ferns, the gymnosperms and most angiosperms, the recognition of leaf gaps is rather uncertain, because in these plants the parenchyma that occurs above the diverging leaf trace is confluent with the interfascicular regions. In such stems, the gaps become delimited only after the addition of some secondary vascular tissue. In plants with vascular bundles depressed within the ground tissue, the delimitation of gaps is even more problematic. Breaking of vascular slender by gaps and reduction. Leaf gaps vary much in width and in longitudinal extent. Their size is not directly related to the size, type or persistence of leaf. Commonly in the gymnosperms and angiosperms, leaf gaps are small, extending but for a short distance above the point where the trace leaves the vascular slender. In the ferns, leaf gaps are generally larger and may extend for considerable distances, even through several internodes. The vascular slender is broken into isolated strands by the presence of leaf gaps. When the gaps are small and with limited longitudinal extent, the slender is slightly broken by them. On the contrary, when the gaps are large and elongate, extending through one or more internode, the stele, that saphenostele, is dissected. In many plants, the primary vascular slender is still further dissected. This additional breakup, which took place in evolutionary development, has been brought about by the progressive reduction in the amount of primary xylem. At first, xylem is reduced radially so that the slender becomes thin. Then it is reduced tangentially so that some longitudinal strips of the slender lost all primary vascular tissue. The non-vascular areas are sometimes called interfascicular rays. The dissection of primary vascular slender by the presence of leaf gaps as well as by interfascicular rays gives it the appearance of network of anastomosing strands. Now, description of nodes on the basis of number of leaf gaps. Depending upon whether 1, 3, 5 or more leaf gaps are left in the stele by the departure of traces, nodes are described as unilacunar, trilacunar, pentalacunar or multilacunar. The nodal pattern is often expressed in terms of the number of traces and gaps. For example, a unilacunar node with a single trace would be described as 1:1. Unilacunar node with two diverging traces would be described as 2:1. And a trilacunar node with a three trace would be described as 3:3, so on and so forth. The first digit denotes the number of traces and the second digit the number of gaps. Most families tend to have uniform nodal anatomy. Variability in nodal anatomy is important and may be of systematic value in intrafamilial classification. Studies show that a unilacunar node with two trace type is present in ginkgophytes and other gymnosperms and some angiosperms. Multilacunar nodes are relatively uncommon in dicotyledonous families, but are present in members of some primitive as well as advanced orders. The unilacunar node is scattered in various groups but can distinguish an entire family, that is Theaceae. As leaves are reduced in size, sometimes as a result of more xeric condition, the nodal anatomy is concomitantly reduced from trilacunar to unilacunar. Although there exists great difficulty in the application of concept of leaf gaps to many vascular plants, still it is commonly utilized in the characterization of nodes. Types of nodes in dicots. 4 main types of nodes are recognized in dicots. Number 1, 2-trace unilacunar with a single gap and 2 trace to one leaf connected to the opposite halves of the axial vascular system. Second, 1-trace unilacunar with a single gap and a single trace to a leaf. Third, trilacunar, that's with 3 gaps and 3 trace to a leaf, 1 median and 2 lateral. And fourth one, multilacunar with several to many gaps and trace to a leaf. However, trilacunar nodes are more common. Now, phylogeny of nodes. According to Sennett, that's 1914, the trilacunar node is the primitive type in the angiosperms, and that the unilacunar node developed phylogenetically from it by the loss of 2 lateral gaps together with their respective traces, or by the approximation of the lateral traces to the median bundles to form a bundle composed of 3 traces, which is associated therefore with a single gap. Contrary to the reduction in the process of formation of unilacular node, Sennett stated that the multilacunar node is formed by the addition of new gaps and traces. However, later research on nodal anatomy of gymnosperms and angiosperms have refuted Sennett's view that trilacunar node is primitive among angiosperms. We have scientists like Grunkel and Wetmore, Fawn, Bailey. These workers and others have shown that in many pteridophytes and in the chordatails, benititails, in Jingo and in Ephedra, a single gap is found in that position where the leaf trace depart from the stele. Similarly, in many dicotyledons, unilacular nodes with 2 leaf trace have also been found. Many of the dicotyledonous genera with unilacular nodes with a double leaf trace belong to the primitive groups of raniales and Ctenopodiaceae. Bailey in 1956 found in many dicotyledons that the vascular sepal to the cotyledons consists of a double leaf trace, which arises from a unilacular node. Unilacular 2-trace node also occurs in many advanced dicotyledonous taxa belonging to Laurels, Verbenaceae, Labiatae and Solanaceae. He is of the opinion that the leaves of the angiosperm are able to undergo reversible changes in shape and vascularization. In the light of these facts, it can be assumed that the unilacular node of certain genera of the raniales is primitive and has not changed during its evolution from that of the lower Pteropsida. Second, in certain other dicotyledonous genera, for example, Leguminaceae and Anacardiaceae and others, the unilacular node has apparently been derived from the reduction of a trilacular node. Third, there are positive indications in certain dicotyledonous groups such as Epiacridaceae and Chloranthaceae that in the course of evolution the tri and multilacular nodes have arisen from the unilacular node. Additional modification with more than 2 traces to 1 gap also appeared. The various phylogenetic changes involved deletions, fusions and addition of traces. The evolutionary development from a unilacular node with 2 leaf trace to other types of unilacular nodes with 1, 3 or more trace may occur in a single family, as can be demonstrated in the Ctenopodiaceae. More than one evolutionary type may be present in the same type. For example, in the Netaceae, Ephedra has unilacular nodes with 2 leaf traces, whereas in Netum, the nodes are multilacunar. The nodal anatomy of Netum represents the highest degree of specialization among the gymnosperms. The nature of leaf trace as well as nodal type is studied only after accurately following the passage of leaf trace downwards in the stem. With this, we concluded today's topic, that was about nodal anatomy, wherein we learned how vascular connections between stems and leaves are maintained through leaf traces and leaf gaps. We also discussed various types of nodes based on the number of leaf traces and leaf gaps and their importance in plant taxonomy and phylogeny. Thank you.